somewhere inside you, written in the quiet architecture of your cells, there is a record that stretches back further than any written history, further than any monument, further than any story passed down around any fire. It is not a story told in words, it is told in nucleotides, in the tiny chemical letters that make up your DNA, and it has been accumulating letter by letter, mutation by quiet mutation, for hundreds of thousands of years. Tonight we are going to follow that record backward. We are going to move slowly, gently through time, back through the centuries, back through the millennia, back into a world that looked and felt almost nothing like the one we know. A world of ice sheets that swallowed continents, of grasslands stretching where cities now stand, of small groups of people walking through landscapes that no human had ever walked before. And we're going to do it not as a race through facts, but as a quiet journey, the kind of journey that asks you to simply rest and let the deep past settle around you like something familiar. The first thing to understand is that the Ice Age was not a single frozen moment. When people hear those words, the Ice Age, they sometimes picture one long unbroken winter, a static scene, as though someone pressed pause on the world, but the reality was far more subtle, far more varied, and in its own way, far more interesting. What we call the Ice Age was actually a sprawling era, technically known as the Pleistocene Epoch, that lasted from roughly 2.5 million years ago until about 12,000 years ago. Within that vast stretch of time, the climate breathed in and out. Glaciers advanced and retreated. Seas rose and fell. Coastlines appeared and disappeared. There were periods of relative warmth tucked inside the cold, called interglacials, where forests grew back and temperatures climbed, followed by periods where the ice surged forward again. The last great maximum of glacial cold, the moment when the ice sheets reached their furthest reach, came roughly 20,000 years ago, and it is that period and the thousands of years surrounding it that left some of the deepest marks on who we are today. The people who lived through those shifting climates didn't know they were living through something that would one day be studied and reconstructed by scientists with sequencing machines and statistical models. They were simply living, finding food, staying warm, moving when the land demanded it, staying when it allowed. But in living, they were also writing. Every time a population shrank, every time a small group wandered into new territory, every time two distant bands met and had children together, that event left a trace, a subtle shift in the frequency of a genetic variant, a new mutation carried forward into all descendants, a pattern in the diversity of lineages that a careful analysis, conducted tens of thousands of years later, can begin to read. This is where the science becomes something genuinely remarkable. Modern genetics, when applied to questions of deep ancestry, works not through single dramatic discoveries, but through the patient accumulation of evidence from multiple directions at once. Geneticists study DNA from living people around the world, looking at how variation is distributed across populations, noting which groups share certain markers and which do not. They also increasingly study ancient DNA, genetic material extracted from bones and teeth found at archaeological sites, from individuals who lived 5,000, 20,000, even 40,000 years ago. These ancient genomes act as direct witnesses. They allow researchers to look back past all the mixing and movement of more recent history and see something closer to the original signal. But genetics does not work alone. The picture it builds is always richer and always more trustworthy when it is read alongside the physical record, the fossils, the stone tools, the traces of ancient campsites and butchered animals and painted caves. Archaeology tells us where people were and roughly when. Paleoclimatology tells us what the environment looked like. Genetics tells us about relationships, about movement, about population size, about the hidden chapters that left no tools behind. Each discipline asks different questions of the past and receives different kinds of answers, and it is only in conversation with one another that they begin to assemble something approaching a coherent story. It is worth being honest at the outset of this journey about what that story can and cannot yet tell us. Genetics is a powerful tool, but it is not a perfect window. There are regions of the world where ancient DNA is difficult or impossible to recover because humid climates degrade genetic material quickly. There are populations whose histories remain deeply uncertain, where the available data is sparse or contested. There are questions about the interpretation of genetic patterns that researchers continue to debate, where the same evidence supports more than one narrative. The picture we have is extraordinary in its detail and ambition, but it is still a picture with gaps, with blurred edges, with regions where the light has not yet reached. None of that uncertainty should discourage us. If anything, it is part of what makes this field feel so alive. We are living at a moment when ancient DNA research is still young, when new discoveries genuinely revise what we thought we knew, when the map of deep human history is being redrawn in ways that would have seemed impossible even 30 years ago. What we do know is already astonishing, and what we are still learning is more astonishing still. Tonight, we will move through this world gradually. We will spend time in the cold landscapes, with the small bands of people navigating them. We will follow the first great movements out of Africa and across unknown continents. We will sit with the strange and affecting story of what happened when modern humans met other kinds of humans, Neanderthals, Denisovans, others perhaps still unnamed. We will look at the quiet ways the climate shaped bodies and genes over generations, and we will eventually arrive back at the present, at the people alive today, who carry all of this within them without knowing it on most ordinary days. For now, let the journey begin gently. Let the ordinary world grow a little quieter, a little further away. Somewhere beneath the noise of the present, your cells hold an ancient record, and tonight we are going to listen to it together. To understand what that ancient record holds, you first need to feel the world it was written in. Not the world of cities and agriculture and permanent settlement, but the world as it existed for the vast majority of human prehistory. A world of extraordinary restless environmental change, where the ground beneath people's feet was never entirely still, and where survival depended on a constant, unconscious negotiation with climate. The Pleistocene Epoch, that long era stretching back more than 2 million years, was defined above all else by cycles. Not smooth, predictable cycles, but irregular ones, driven by subtle variations in the Earth's orbit and tilt, by the way sunlight fell across the northern hemisphere over tens of thousands of years. These orbital rhythms, named Milankovitch cycles after the mathematician who worked them out, cause the planet's climate to oscillate between cold glacial periods and warmer interglacials in a slow grinding rhythm. The ice would grow, the ice would retreat, then it would grow again. Over the course of the Pleistocene, this pattern repeated roughly 20 times, each cycle lasting somewhere between 80,000 and 100,000 years in its fullest expression. During the cold phases, ice sheets of almost incomprehensible scale spread outward from the poles. At the last glacial maximum, around 20,000 years ago, ice covered much of northern Europe. Scandinavia disappeared entirely beneath a dome of glacial ice more than 3 kilometers thick in places, and the ice sheet over North America reached as far south as what is now the northern United States. The weight of all that ice was enough to press the land itself downward, deforming the crust of the Earth. And because so much of the planet's water was locked up in glacial ice, sea levels dropped, by some estimates by as much as 120 meters below where they stand today. Coastlines that are now submerged under ocean were once dry land, sometimes vast stretches of it, and those exposed shelves and plains were among the most important stages in the human story. What this meant for the landscape was transformation on a scale that is difficult to fully imagine. Forest retreated, tundra expanded. The great open grasslands that ecologists sometimes call the Mammoth Steppe extended across enormous portions of Europe, Asia, and into the Americas, a connected belt of cold, dry, wind-swept vegetation that supported large herds of grazing animals, woolly mammoths, woolly rhinoceroses, cave lions, bison, horses, reindeer. This steppe tundra environment was not lifeless. In ecological terms, it was actually highly productive in its own way, sustaining animal populations of considerable density. For human groups who had the tools and knowledge to hunt there, it offered real opportunity. But it was also harsh, bitterly cold in winter, and unforgiving to those without the right adaptations. Not all regions experienced the ice ages in the same way. Proximity to ice sheets mattered enormously, as did latitude, altitude, and local geography. Certain regions remained relatively mild even during the coldest periods, places protected by their position, or warmed by ocean currents, or sheltered by mountain topography. These refuges were vital. When ice advanced, plants and animals were pushed back toward these more hospitable pockets, and human populations moved with them. Refuges in southern Europe, in parts of Central Asia, along the coasts of Africa, in pockets of the Middle East. These became places where life concentrated, where populations held on during the worst of the cold, and from which recolonization could begin when the ice retreated. The sea level changes that accompanied glaciation were just as consequential as the cold itself. When so much water was locked in ice, the shallow continental shelves became dry land. The region we now call Sunderland, the shallow sea floor connecting the islands of Southeast Asia, was once a substantial landmass, a broad low plain extending well beyond the current coastlines of Indonesia, Malaysia and Indochina. Beringia, the land bridge connecting northeastern Asia to northwestern North America, was not a narrow strip but a wide windswept subcontinent in its own right. These land bridges and exposed plains opened pathways that did not exist before and would not exist again, creating corridors through which both animals and people could move into territories that were otherwise inaccessible. When the ice retreated and the seas rose, those corridors closed. Populations that had crossed became separated by water from the places they had come from, isolated in ways that would shape their genetic stories for thousands of years afterward. Between the cold periods came the interglacials, and these brought their own transformations. Temperatures rose, sometimes to levels warmer than today. Forests expanded northward and uphill. The great grasslands shrank back, replaced by woodland and then by denser forest. For human populations adapted to open steppe-like environments, this contraction could be as challenging as the cold itself. The prey animals they had organized their lives around moved or declined. The open landscapes they had navigated closed over, and new challenges replaced old ones. Adaptation was never a one-time achievement. It was an ongoing process, a continual renegotiation with the shifting terms offered by the environment. What is perhaps most important to understand is that this instability was not background noise. It was the central fact of human experience for most of the species' existence. The relative climatic stability of the last 12,000 years, the warm, mild Holocene that made agriculture possible and cities imaginable, is the exception. The Ice Age was the rule. For hundreds of thousands of years, human populations lived inside a world that was always, on some timescale, in the process of becoming something different, and that world selected quietly and persistently for particular qualities, for flexibility, for the ability to move when movement became necessary, for the capacity to learn new landscapes and new food sources and new strategies with each generation that found itself living somewhere their grandparents had not. Migration, under these conditions, was not an adventure or a choice in any simple sense. It was often the consequence of pressure, of ice advancing, of prey moving on, of a local landscape becoming unable to support the people who depended on it. Small groups followed the margins of the habitable world, pressing into new territories not because those territories called to them, but because the territories behind them had changed. Isolation, just as often, was the consequence of the opposite, of populations being cut off by rising seas, by expanding ice, by the closing of corridors that had briefly been open, and that alternation between contact and isolation, between mixing and separation, between expansion and contraction, is precisely what gave rise to the patterns geneticists can now read in human DNA. The physical world of the Ice Age was also a world of profound local diversity. A valley in what is now southern France might have been a relatively sheltered environment, supporting a small human population through the worst of the cold, while 50 kilometres to the north, conditions were close to uninhabitable. A coastal refuge in southern Africa might have offered shellfish and calm weather, while the interior was grassland locked in drought. People did not experience climate change as a global phenomenon. They experienced it as the particular landscape in front of them, the specific animals available, the specific plants that grew, the specific winters they survived, and the ones they did not. It was local adaptation, again and again, in thousands of different places that accumulated into the diversity we can trace today. Let this world settle around you for a moment. Ice pressing down from the north, seafloors exposed to open air, grasslands stretching for thousands of kilometres under cold, pale skies. Small groups of people moving through all of it, carrying with them fire and tools and knowledge and the invisible cargo of their genes. This was the stage. The next question is who was living on it, and what it meant in practice to be human inside that cold, shifting, demanding, and endlessly changing world. Picture a group of perhaps 20 or 30 people moving across an open landscape sometime near the height of the last glacial maximum. There are children among them, and elderly members who carry knowledge accumulated over decades of navigating this terrain. There are hunters and those who gather roots and tubers and seeds and insects and anything else that offers nutrition. There is a fire or the means to make one. There are tools, carefully shaped stone points, scrapers, needles carved from bone, that represent accumulated skill passed from one generation to the next. And there is a web of relationship binding all of these people together, a network of kinship and obligation and shared memory that is, in many ways, the most important survival technology they possess. This was the fundamental unit of human life throughout the Ice Age. Not the tribe in any large or abstract sense, but the band, a small, flexible, mobile group of closely related and socially bonded individuals who move through a defined territory according to the rhythms of the season and the availability of food. Archaeologists and anthropologists who study both the archaeological record and the practices of hunter-gatherer societies that survived into the modern era tend to converge on similar numbers. Bands were typically small, often between 15 and 50 individuals, rarely much larger for any sustained period of time. They were intimate communities in the most complete sense. Everyone knew everyone. Dependencies were direct and personal, and the loss of even a few members could threaten the group's viability. Seasonal movement was the rhythm that organized everything else. In spring and summer, when plant foods were more available, and certain animals moved to particular grazing areas, bands might spread out across a wider range, taking advantage of abundance where they found it. In winter, resources concentrated, mobility became harder, and groups tended to pull back toward more sheltered locations where fuel and food could be more reliably found. This seasonal pulsing, expansion and contraction, dispersal and return, created a pattern of movement that was neither random wandering nor fixed settlement, but something more structured and purposeful. A seasonal round, repeated year after year with variations imposed by changing conditions. Hunting during the Ice Age required remarkable coordination. Large game animals, mammoths, bison, horses, reindeer, were not easy prey for small groups of humans carrying stone-tipped spears. Successful hunts often depended on collective strategy, driving animals toward natural features that limited their escape, surrounding smaller prey on open ground, exploiting seasonal patterns of migration to intercept herds at predictable crossing points. This kind of hunting was inherently social. It required shared planning, trust, and a division of roles that assumed each person would do their part. The bands that could organize this cooperation most reliably were the ones that ate regularly. Gathering complemented hunting in ways that archaeological evidence increasingly confirms were significant. Plant foods, insects, shellfish along coastlines, freshwater resources, eggs and tubers and nuts and berries when available. These were not incidental additions to a diet centered entirely on meat. They were essential contributions that varied by season and location, and that required their own specialized knowledge. Knowing which plants were edible and when, which required processing before they could be consumed, which were dangerous and in what doses, this knowledge was not trivial. It was accumulated over lifetimes and shared within the group, another form of inheritance passed alongside genes. What matters for the genetic story is not just what these bands did, but how isolated they were from one another. Low population density, across a landscape as vast and challenging as Ice Age Eurasia or Africa, meant that bands often went long periods without encountering other human groups. When the territory between two groups was hundreds of kilometers of cold steppe or dense forest or semi-desert, regular contact became difficult and sometimes impossible. Births and deaths, and the random outcomes of who survived to reproduce, what geneticists call genetic drift, had an outsized effect in small isolated populations. In a group of 30 people, a single bad winter that killed several adults of reproductive age could meaningfully shift the genetic composition of every generation that followed. Traits and variants that were common in one group might be entirely absent in the neighbouring group over the next ridge, not because of any adaptive pressure, but simply because of the accumulating weight of chance in small numbers. This is genetic drift in its most intimate form, not an abstract statistical process, but the lived consequence of small, scattered communities facing an uncertain world. When a band was isolated for many generations, its genetic profile became increasingly distinct from other bands, shaped by its own particular accidents of survival and reproduction. And when two bands finally met and exchanged members, through the marriage alliances and reciprocal relationships that are a universal feature of hunter-gatherer social life, those distinct profiles began to blend again. The history of human genetic diversity is, in large part, the history of that alternating rhythm. Separation deepening difference, contact blending it back together. Family networks were not separate from survival strategies. They were the same thing. The connections maintained between neighbouring bands, through marriage, through gift exchange, through shared ceremonies and mutual obligation, were a form of insurance. When one group's territory failed to provide enough food, they could call on the relationships they had maintained with others to gain temporary access to a different range. When a band lost too many members to sustain itself, individuals or small family groups might be absorbed into neighbouring bands. When a band grew large enough to become unwieldy, it would split, with one portion moving off to establish a new range while maintaining ties to the original group. These movements of individuals and small family units, driven by the social logic of kinship and alliance, were as important to the shaping of human genetic geography as any large migration event. Who reproduced and with whom was rarely a purely individual matter. Across hunter-gatherer societies, marriage arrangements involved the whole community, with kinship rules governing which partnerships were permitted and which were not, and with social pressures guiding who ended up together and where they settled afterward. In many band-level societies, marriage involved one partner, usually though not always a woman, moving from her birth group to live with her spouse's group, or some variation of this pattern. Over generations, this practice meant that certain genetic lineages flowed preferentially in particular directions, carried along social pathways that were themselves shaped by geography, alliance, and opportunity. The genetic signatures left by Ice Age bands are, in this way, also social signatures, records of who was connected to whom, and how those connections were maintained across the cold and difficult distances of the Pleistocene landscape. What is striking, looking back, is how completely ordinary all of this was. The people living inside these patterns were not thinking about genetic drift or population structure. They were thinking about where the reindeer had gone, about whether the frost had damaged the root beds they had returned to count on, about who among the young people of the neighbouring band might make a good partner for a daughter reaching adulthood. They were managing cold and hunger and childbirth and illness and grief with the tools and knowledge they had. Survival was the immediate concern, and the long-term genetic consequences were simply what accumulated from thousands of those immediate, concrete, utterly human decisions. The genetic record that scientists can now read from ancient and modern DNA is, in a very real sense, the sediment left by all of those daily lives. Each small choice about where to move, whom to marry, whom to shelter and care for and feed through a hard winter, each of these contributed a tiny increment to the pattern that would still be legible tens of thousands of years later. The isolation of a band during a particularly severe cold period, the absorption of a few individuals from a distant group following a chance encounter, the founding of a new band at the edge of explored territory by a small group that carried only a fraction of the genetic diversity of the parent population. These were not dramatic events. They were the texture of ordinary life in a vast, cold, sparsely peopled world. And that texture, repeated endlessly across millennia, is what shaped the deep architecture of human genetic diversity that persists today. Somewhere between 60 and 80,000 years ago, something shifted. The small bands that had lived for hundreds of thousands of years within the African continent began, in some cases, to move beyond it. Not in any coordinated sense, not as migration in the way we might imagine armies or caravans moving with purpose toward a known destination. The departures were gradual, probably driven by the same quiet pressures that had always pushed human bands across landscapes. The search for reliable food, the movement away from drought or competition, the slow accumulated drift of seasonal rounds that, over generations, carried people farther than any individual lifetime would have registered. And yet the consequences of those unhurried movements were enormous, shaping the genetic geography of the entire species in ways that are still visible in every living person on Earth today. The timing and the routes of this dispersal remain genuinely debated among researchers. Genetic evidence from both ancient and modern populations suggests that most of the ancestry of people living outside Africa traces back to a relatively small founding population that left the continent sometime during this broad window, moving through what is now the Horn of Africa, or the Sinai Peninsula, or possibly both. But the picture is complicated by older findings, archaeological sites in Arabia, tools in Australia, and fragments of ancient DNA that hint at earlier movements, earlier explorations that may have left little or no trace in living populations. Some of these earlier departures may have died out, populations stranded by climate shifts, or absorbed into later waves without leaving a detectable signature. Others may have contributed small amounts of ancestry that are only now becoming visible as ancient DNA sequencing improves. What can be said with confidence is that the major genetic lineage ancestral to nearly all non-African people today appears to share a relatively recent common origin, suggesting that even if earlier groups ventured out, the enduring story of human expansion began with a comparatively small number of founders. Climate played a decisive role in when and how movement was possible. The period between approximately 60 and 50,000 years ago included intervals when sea levels were lower than today, exposing shallow coastal shelves that no longer exist, and creating land bridges or narrow crossings that would have made passage into Arabia and beyond more feasible. The Bab el-Mandeb Strait at the southern end of the Red Sea, which today separates East Africa from Yemen by about 30 kilometres, would have been significantly narrower during periods of glacial sea level reduction, possibly narrow enough for small groups to cross on simple watercraft, or even to wade across exposed shoals. Once on the Arabian side, the landscape was not the desert it is today. During wetter intervals driven by monsoon patterns, the Arabian Peninsula supported grasslands and water sources that could have sustained hunter-gatherer bands moving eastward. The coastal route hypothesis has attracted considerable attention. Moving along shorelines offered consistent access to marine and coastal resources, shellfish, fish, seabirds, the predictable productivity of intertidal zones, in ways that inland routes might not have guaranteed. A band moving along a coastline could travel enormous distances over generations while remaining within a familiar ecological niche, never venturing too far into unknown territory at any single step. The genetic and archaeological evidence does not definitively confirm this route over others, but the idea fits well with what is known about the speed and direction of early dispersals. Populations appear to have reached South Asia, Southeast Asia and Australia with remarkable rapidity in evolutionary terms, within a few thousand years of the initial departure from Africa, a pace more consistent with coastal movement than with slow inland migration through varied and often difficult terrain. Inland corridors also played a role as conditions permitted. The Nile Valley, the Levant and the grassland corridors that periodically opened through Arabia and into the Iranian plateau offered pathways that expanding bands could follow when climate windows aligned. These routes were not always open. Periods of increased aridity could close them for millennia, isolating populations that had already moved through and preventing further contact with groups still in Africa. This alternating pattern of connection and isolation, corridors opening and closing with climate cycles, created a complicated layering of movements that geneticists are still working to disentangle. As groups travelled and split, something consequential happened to their genetic diversity. The founders of each new population carried only a sample of the variation present in the group they came from. When a small band departed from a larger parent population and established itself in new territory, it brought with it whatever genetic variants its particular members happened to carry, not the full range present in the community left behind. This narrowing is called a founder effect, and it is one of the most consistent signatures of the out of Africa dispersal. Populations that settled farthest from Africa tend to carry the least genetic diversity, a pattern so reliable that the genetic diversity of a population correlates roughly with its geographic distance from the African continent even today. Each successive split and movement filtered the variation a little further, so that populations in East Asia or the Americas carry a subset of the diversity found in South Asia, which itself carries a subset of what exists in Africa. This is not a story of loss in any tragic sense. The variants that travelled with the founders were real and functional, and the populations that descended from them were fully capable of adapting to the environments they entered. But the statistical fingerprint of those ancient bottlenecks, the moments when small groups set off and left most of the parent population behind, is still embedded in genomes today. Scientists can identify these signatures, estimate how small the founding populations were, and sometimes infer roughly when the separations occurred by measuring how long the distinct genetic patterns have had to accumulate. The out-of-Africa founders may have numbered only in the hundreds or low thousands of individuals at the point when they became genetically separated from African populations, a remarkably small number given that they are the ancestors of billions of people now living across every continent. Migration did not happen in a single wave. The genetic evidence increasingly supports a picture of multiple movements, some overlapping, some separated by thousands of years, some reaching the same regions from different directions at different times. An early dispersal may have carried modern human ancestry deep into South and Southeast Asia, before a separate movement populated Europe and Central Asia. Later expansions reshuffled the earlier patterns, with some populations expanding dramatically and others contracting or disappearing entirely. The waves do not cancel each other out in the genome. They layer, leaving composite signals that require careful analysis to distinguish. A person in South Asia today may carry ancestry from several distinct founding events, each visible as a different component of their overall genetic profile, each traceable to a different chapter of these early departures. What is easy to lose sight of following these abstract patterns of dispersal and founder effects is that every movement was made by actual people who had no awareness of contributing to a species-wide expansion. A small group pressing slightly beyond the range their parents had known, following the edge of a receding coast or crossing a shallow strait on a raft of bound reeds, was simply doing what bands had always done, seeking territory that could sustain them, moving with the rhythms of food and season and the press of other groups behind them. The genetic consequences, the narrowing of diversity, the isolation that would deepen difference over thousands of generations, the founding of lineages that would eventually number in the billions, were invisible and unimaginable to the people living through them. They were the long echo of ordinary movement accumulating silently into history. The departures from Africa were not an ending. They were the beginning of a vast and still unfolding diversification, the first chapters of a genetic narrative that would eventually include the peopling of every habitable corner of the planet, the accumulation of adaptations suited to deserts and Arctic coastlines and tropical forests, the blending of lineages through encounters that were sometimes friendly and sometimes violent, and sometimes simply the slow, gravitational pull of proximity. All of it began with small bands moving across familiar landscapes, gradually, without fanfare, carrying in their bodies the seeds of every population that would follow. As some of these bands of Homo sapiens moved farther from the African homeland, they entered regions that had not been empty. Long before our particular lineage crossed those narrowing straits and grassland corridors, other humans had already spread out into Eurasia. The world they were entering was not just a mosaic of climates and animals, but a mosaic of people. In the colder reaches of Europe and Western Asia lived the Neanderthals. They had been there for hundreds of thousands of years, weathering multiple swings between glacial cold and milder interglacial periods. Their bones tell of barrel-shaped chests, strong limbs, and faces adapted to the chill. But in the simplest sense, they were humans too. They made stone tools, controlled fire, hunted large animals, and likely cared for their injured and dead. Farther east, in the mountains of Central and Eastern Asia, another more shadowy branch of the human family persisted. The Denisovans, known at first only from a few fragments of bone and teeth found in a Siberian cave, and later from the ghostly traces of their DNA. For a long time, The relationship between these ancient humans and ourselves was imagined as a succession. First came earlier species, then Neanderthals, and finally modern humans, replacing all who came before. It was a clean, tidy model, appealing in its simplicity, one species following another, each step more modern than the last. But genomes do not tell tidy stories. When scientists first managed to extract and sequence DNA from Neanderthal bones, a different picture began to emerge, one that quietly overturned the idea of complete replacement. If modern humans and Neanderthals had diverged and then lived entirely separate lives, their DNA should have been cleanly distinct, with no sign of shared genetic material beyond what both inherited from older common ancestors. Instead, when the first high-quality Neanderthal genome was compared to the genomes of people living today, a faint but unmistakable signal appeared. Individuals whose ancestors lived outside Africa carried small stretches of DNA that matched the Neanderthal sequence more closely than they matched that of any living African population. These sequences were not scattered randomly. They formed recognizable blocks, broken up by recombination over tens of thousands of years, but still large enough to stand out. The simplest explanation was also the most profound. At some point after Homo sapiens left Africa, they met Neanderthals and had children together. Those children grew up, had their own children, and some of their descendants are alive today. The traces of those ancient matings are still present as tiny segments, a few % of the genomes of most people with ancestry in Europe, the Middle East, and much of Asia. The old picture of a single, pure line of modern humans sweeping aside earlier forms without mixing could not survive this evidence. Our ancestry, it turned out, was not a straight line but a braid. A similar story unfolded for the Denisovans. At first, their identity was known only from their genetic sequence, recovered from a small finger bone in Denisova cave. To the surprise of researchers, this DNA was clearly neither Neanderthal nor modern human, but a different branch, splitting off earlier in the Eurasian story. When this Denisovan DNA was compared to the genomes of people living today, the echoes appeared again. Populations in parts of East and Southeast Asia, and especially in the Pacific, in places like Papua New Guinea, parts of Melanesia, and among some indigenous Australian groups, carried distinctive stretches of DNA that matched the Denisovan genome more closely than any other. Once again, the conclusion was clear. As Homo sapiens moved into these regions, they did not simply displace the humans they found there. They met them, lived near them, and in some cases had children with them. The timing of these encounters can be sketched in broad strokes. Neanderthals occupied Europe and Western Asia until roughly 40,000 years ago, with their range reaching into regions that newly arriving Homo sapiens also entered. The Levant, parts of Central Asia, the corridors leading into Europe. The interbreeding events that left the clearest traces in present-day genomes likely occurred early in this overlap, perhaps around 50 to 60,000 years ago, when modern humans were first expanding through the Middle East and beyond. The Denisovan encounters may have stretched over a wider span in multiple regions, from the highlands of Central Asia to the islands of Southeast Asia, as human groups moved along coasts and through mountain passes into new lands. These were not singular dramatic episodes, but likely a series of localized, ordinary human interactions. Small groups meeting at the edge of territories, exchanging information, sometimes competing, sometimes forming alliances, and occasionally forming families that crossed whatever boundaries language and appearance might have created. The individuals involved did not know they were representatives of different species. They were simply people encountering other people, judging one another by the same cues humans have always used: behavior, trust, shared survival. The genetic legacy of these meetings is not distributed evenly around the world. People whose recent ancestry lies entirely within sub-Saharan Africa generally carry very little or no detectable Neanderthal or Denisovan DNA, because their ancestors' lineages did not pass through the regions where these other humans lived. By contrast, many populations in Europe, the Middle East and much of Asia carry around 1 to 2% Neanderthal ancestry on average, though the exact amount varies. Some populations in Oceania and nearby regions carry a noticeable share of Denisovan ancestry, in addition to Neanderthal traces. East Asian populations, intriguingly, often show slightly higher average Neanderthal ancestry than Europeans, hinting that there may have been multiple episodes of interbreeding in different places and times. These patterns form a map of ancient contact zones, drawn not on stone or parchment, but in the mosaic of our genomes. They show, for example, that the people living today in Siberia and Tibet do not share the same Denisovan legacy as those in New Guinea and parts of Melanesia. The segments they carry come from different branches of the Denisovan family tree. This suggests that modern humans did not meet a single, uniform Denisovan population, but different groups, separated by distance and time, each with their own deep histories. What emerges from all of this is a sense of human evolution as a braided river. Branches split from a main channel, flow separately for a while, then rejoin, mixing waters before splitting again. Our lineage within Homo is not a straight ascent. It is a tangle of diverging and reconnecting lines. Neanderthals and Denisovans were not failed experiments neatly replaced by a superior model. They were our close relatives, part of a shared landscape of humankinds that overlapped and interacted across Ice Age Eurasia. The fact that their DNA persists in us means that, in a small but real way, they did not vanish entirely when their distinctive skeletons disappear from the fossil record. Pieces of their genomes still circulate in living humans, recombined and diluted over thousands of generations, but present all the same. Each of us outside Africa carries a small genetic reminder that our ancestors did not journey alone into the wider world. They travelled through lands already peopled, lands where, under cold skies and shifting climates, different humans met, recognized one another as potential partners, and wove their stories together. For the purposes of sleep, there is a quiet reassurance in this braided past. The borders between us and them, drawn so sharply in modern imagination, blur when we look back far enough. Where our ancestors walked, they encountered others who were different and yet fundamentally similar. Fire-making, tool-crafting, family-forming humans. The story of our species is not a solitary thread, but a bundle of threads, twisted together by countless small acts of contact. In your own DNA, some of those threads still carry the faint signature of Neanderthal valleys and Denisovan mountains, reminders that human history has always been, in the deepest sense, shared. Across these woven lineages, something else was working slowly, almost invisibly in the background. It did not care about names or borders, or which branch of the human family a person belonged to. It cared only about who survived long enough to have children and who did not. This quiet force was natural selection, and in the deep chill of the Ice Age it had a particular set of questions to ask of every body that walked under those pale skies. Cold, darkness, scarce food, and long winters pressed in on all human groups living beyond the tropics. None of these pressures were sudden in the way a storm is sudden. They unfolded over thousands of years, over the rise and fall of glaciers, over the slow advance of forests and the retreat of grasslands. But within that slow movement, every individual still faced very immediate challenges: not freezing in the night, not starving between hunts, not losing too much heat or too much water, not becoming too weak to care for their children. In a small band of people, the differences between individuals might be subtle. One person might be slightly better at keeping warm on little food, another might tire less quickly when walking long distances in the snow, another might heal a bit faster after frostbite. No one would have words for the inner workings of these differences, yet over many generations they mattered. Those who, by chance, had combinations of traits that worked well in that particular patch of Ice Age land left a few more surviving children. Their genes, in turn, became a little more common in the group. This is the essence of selection in small populations. The changes from one generation to the next are tiny, almost imperceptible. In any single lifetime, the cold sky looks much the same. But from a vantage point of tens of thousands of years, small biases in survival and reproduction add up. In one valley, where winter wind is constant and food is often lean, certain body shapes and metabolisms gradually become the norm. In a more sheltered coastal region, with access to marine foods and slightly milder temperatures, a different balance of traits ends up favoured. Nature is not aiming at a perfect form. It is simply keeping what works slightly better than the alternatives, over and over across deep time. The cold did not act alone. Darkness was its constant companion at high latitudes. In those long, dim winters, people spent many weeks with little or no direct sunlight. That meant their bodies had to juggle several demands at once, maintaining body temperature, balancing sleep and wakefulness in the absence of strong day-night cues, and managing the chemistry that sunlight normally helps with, like the production of vitamin D. Diet played a role here too. Groups who could harvest fat-rich fish or marine mammals, or who followed herds that provided nutrient-dense meat and marrow, had different options than those relying on seasonal plant foods and smaller game. All of these pressures intersected in the same place, the human body's ability to regulate energy. To stay alive in the cold, a body must constantly decide what to do with the calories it has. Should energy be used to move, to think, to repair damaged tissue, to grow, to reproduce, or simply to keep the core of the body from dropping a few dangerous degrees? The solutions that emerged over thousands of years involved an intricate choreography of fat metabolism, circulation, and heat production. Some human groups evolved tendencies towards storing energy in ways that were particularly helpful in icy climates. Certain forms of fat, often called brown or beige fat, are more active in burning calories to generate heat rather than simply storing them. In some modern populations with histories in cold regions, researchers see hints that genes involved in this kind of thermogenic fat have been shaped by past selection, adjusting how easily the body can switch from storage to burning when the temperature drops. The arrangement of blood vessels, too, can make a difference. How quickly fingers and toes are allowed to cool to protect the core, how efficiently warm blood is recirculated, how tightly the body constricts vessels under the skin to reduce heat loss. Circulation patterns and body proportions work together to influence the balance between staying warm and staying mobile. Shorter limbs and more compact torsos reduce exposed surface area and conserve heat, a pattern seen in Neanderthal skeletons and in some modern Arctic and subarctic populations. But no single measurement can tell the whole story. For every person whose body handles cold well, there are dozens of subtle genetic variants interacting, in hormones, in the nervous system, in the muscles, in the way cells take up and burn fats and sugars. This is one reason why the story of adaptation is rarely simple. There is a temptation, especially when we look at genetic data, to search for single, cold genes or neat explanations. One mutation that made people winter hardy, one borrowed Neanderthal variant that suddenly allowed survival in the snow. The evidence we have does not usually support such clean tales. Instead, we see many small changes spread across the genome, each contributing a fraction of the overall effect, tangled together with traits that have little to do with temperature at all. When scientists scan the genomes of people with long histories in cold environments, they sometimes find regions that seem to have been favoured by selection. These regions may include genes related to fat metabolism, to insulin signalling, to the regulation of blood vessels, or to the function of mitochondria, the tiny power plants inside cells. Curiously, some of these same genes are also linked in modern medical studies to conditions like obesity, type 2 diabetes, or heart disease. This does not mean that those diseases were selected for. Rather, it suggests that traits that once helped people manage scarce food and bitter cold can, in today's very different environments, contribute to health problems. Here, it becomes important to distinguish between strong evidence and reasonable speculation. When a particular genetic variant is both common in a population and clearly associated with a cold-relevant trait, say, the ability to endure low temperatures without shivering, or a distinctive pattern of fat use during fasting, and when its rise in frequency lines up in time with known climatic shifts, then the case for adaptation is on firm ground. In a few instances, we can even see that such variants likely came from archaic humans like Neanderthals or Denisovans, and then spread because they were useful. Much more often, however, the signals are faint and the connections indirect. A gene may show signs of selection, but its exact effect on the body is unclear. A trait, like body shape or average metabolic rate, may differ between populations, but the genetic underpinnings are scattered across hundreds of positions in the genome. In such cases, scientists can suggest hypotheses. Perhaps this difference relates to cold stress or to seasonal famine, but they must also acknowledge uncertainties. The map from DNA to lived experience is not yet complete, and in some areas it may never be fully drawn. What is becoming increasingly clear is that many of the traits that mattered for survival in the Ice Age are polygenic. That word simply means involving many genes. Height is a familiar example today, not governed by a single switch, but by thousands of genetic variants, each nudging a person a millimetre taller or shorter. The same is true in its own way for cold tolerance, for metabolic flexibility, for blood pressure responses to stress, for the fine-tuned dance of hormones that tell the body when to store fat and when to release it. Natural selection in small Ice Age bands did not sculpt one gene at a time, like a craftsman carving a single ornament. It worked more like a tide moving over a beach full of pebbles, shifting the shape of the shore in small increments, generation after generation. A tiny change here, a tiny change there, across thousands of individuals and thousands of years, until a particular combination of traits, slightly thicker insulating fat, a marginally more efficient way to burn fuel, a slightly more conservative pattern of blood flow in the cold, became common not because it was perfect, but because it was good enough, often enough in that place. As the braided river of human ancestry flowed through Neanderthal valleys, Denisovan mountains and the open steppes of Ice Age Eurasia, this quiet selection never stopped. It shaped not just faces and bones, but the invisible thermometers and furnaces inside every body, and even today, under electric lights and heated roofs, those ancient adjustments are still with us, playing out in how we feel on a winter morning, in how our bodies respond to a skipped meal, in the silent, constant work of keeping warm and alive. In the next quiet stretch of this story, the attention will turn from the deep core of the body outwards to the surface, to the thin interface between warm blood and cold air. There, under northern skies tinged with weak sun, another slow negotiation with the environment was underway, written not in fat and circulation, but in the colours and chemistries of human skin. Skin is often the first thing we notice about another person. For our Ice Age ancestors, though, it was not a sign of identity or belonging in the way we might imagine today. It was, above all, a working surface, a shield, a filter, and a chemical factory under a changing sky. As some human groups drifted away from the bright direct light of the equatorial sun and into higher latitudes, the relationship between their skin and their surroundings began to change. Under tropical skies the sun is strong and steady through the year. Nearer the poles, it is not. Light arrives at an angle spread thin across the atmosphere. Winters become long tunnels of dimness, and even in summer the sunlight is softer and more slanting. The same body, standing in these different worlds, faces different questions from the environment. One of those questions is how to handle ultraviolet radiation. Ultraviolet, or UV, is the portion of sunlight that can damage DNA, trigger skin cancer, and destroy the delicate molecules of folate in the blood. Folate is vital for many processes, including the proper development of embryos. In bright, high UV environments, a strong, dark layer of pigmentation acts like a natural sunscreen, absorbing and scattering much of this damaging light before it can harm the deeper tissues. Over long stretches of time, in lineages that lived under intense equatorial sunlight, natural selection favoured skin that could reliably provide this protection. Yet ultraviolet light is not only a threat, it is also a crucial ingredient in one of the body's quiet chemical routines, the production of vitamin D. In the upper layers of the skin, UV radiation helps transform a cholesterol-based compound into a precursor of vitamin D, which the liver and kidneys then convert into its active form. Vitamin D in turn supports bone growth, immune function, and many other aspects of health. Thus, the same rays that can damage DNA are also needed to keep skeletons strong and immune systems balanced. In the deep tropics, this tension between protection and production can often be resolved by heavy pigmentation. The sun is so intense and exposure so regular that even with strong melanin shields, enough UV typically penetrates to synthesize adequate vitamin D. But as people moved under northern skies, that balance shifted. The further from the equator they went, the fewer hours per year the skin received usable sunlight, and the weaker that sunlight became, especially in winter. Imagine a small band of hunters on the steppe, or in a patch of tundra forest, where the sun lingers low on the horizon for much of the year. Even in summer, clothing made from animal hides shields much of the body from wind and cold. Faces and hands may be uncovered, but the rest of the skin is often wrapped. In these conditions, the body's opportunity to make vitamin D from sunlight becomes very limited. Eating vitamin D-rich foods, like the fat and liver of marine mammals or fatty fish from coastal waters, helps. But inland groups, depending more on land animals and seasonal plants, might find that diet alone could not always fill the gap. Over thousands of years, these northern circumstances gently tugged on pigmentation patterns. Individuals whose skin let in just a little more UV during brief periods of sun could produce somewhat more vitamin D. If that extra supply meant stronger bones in childhood, fewer rickets-like deformities, or healthier pregnancies, those small advantages would be reflected in survival and fertility. In this quiet way, selection could favour lighter pigmentation where UV was scarce, not because light skin was somehow better, but because in that particular environment, with that particular diet and climate, it solved the vitamin D problem more reliably. This did not mean that there was a simple, single, light skin gene that appeared one day and swept across the north. The reality was far more intricate. Pigmentation is controlled by many genes, each influencing how melanin is produced, packaged, moved, and arranged in the skin. In different parts of the world, different combinations of changes in these genes contributed to lighter or darker tones. Some variants that influence lighter skin in Europe, for example, are distinct from those that play a similar role in parts of East Asia. In other words, similar solutions, more UV passing through the skin in dim climates, were reached through multiple, partly independent genetic pathways. Likewise, darker skin tones in high UV regions did not arise only once and then spread to everyone living under strong sun. They too represent a mosaic of overlapping genetic roots, adjusted and readjusted as populations separated, mixed, and adapted over time. The Ice Age world was not divided into a handful of sharply bounded races with fixed innate packages of traits. It was a patchwork of small groups, each with its own history of movement, diet, and sunlight, gradually shaped by the conditions they encountered. Seasonality added another layer of complexity. At high latitudes, UV is not just weak overall, it is strongly concentrated in a short summer window. For months, there may be almost no meaningful UV at all, followed by a burst of stronger radiation when the sun finally climbs higher. A body living in such a place must make the most of that narrow window. Pigmentation, then, is not simply about how much melanin to have, but about how that melanin is arranged and how responsive the system is to changing exposure. Some modern populations show tanning responses that can dramatically increase pigmentation in summer while remaining much lighter in winter, hinting at a flexible strategy. Protect when you must. Open the gates when you can. Diet and lifestyle also interacted with these forces. Coastal peoples eating large amounts of vitamin D-rich fish and marine mammal fat might face less pressure to lighten their skin, because their food provided what their dim skies did not. Inland groups, with less reliable dietary sources of the vitamin, would lean more heavily on cutaneous production and thus be more sensitive to changes in UV access. In such ways, the story of pigmentation cannot be separated from the story of what people hunted, gathered, and preserved through the long winters. It is important in listening to this long, quiet negotiation between skin and sunlight, not to let modern ideas of race colour the past. The gradients of pigmentation that we see today, darker near the equator on average, lighter in many high-latitude regions, are the result of many overlapping episodes of migration, mixture, and adaptation, not distinct ancient categories. In the Ice Age, boundaries between groups were porous. People met, intermarried, and shared genes just as they shared tools and stories. A single band might carry ancestry from multiple regions, a mixed record of journeys through different climates and under different skies. Even within a single region, change was gradual. No person woke one day to find their community transformed in colour. Over hundreds of generations, a slightly lighter average tone might become common in a valley where winters were long and dietary vitamin D was scarce. In a neighbouring coastal community, with different foods and perhaps slightly stronger sun reflected off the water and ice, that shift might be slower or take a different genetic path. Variance within groups remained large, and individuals spanned a spectrum of shades. The genetic studies that illuminate these patterns today often work backwards, comparing ancient DNA from bones and teeth with the genomes of living people. They reveal that traits like light skin in Europe became common relatively late, after the coldest peak of the last ice age, and that some early farmers and hunter-gatherers in the same region carried very different pigmentation profiles. They show that similar lightening trends occurred in East Asia, and perhaps in parts of the Americas, but with at least partly distinct molecular machinery each time. These findings underline a simple truth. There is no single story of skin colour even in the north. There are many stories, branching and reconverging, all shaped by the same underlying physics of light and biology. Under those pale, angled Ice Age suns, skin was doing quiet arithmetic. How much to shield, how much to let through, how to balance the dangers of UV against the need for vitamin D, all within the constraints set by diet, clothing, and season. The diversity of human pigmentation today is one of the visible legacies of that long balancing act. It is not a map of worth or capacity, but a record of countless generations responding to the simple fact that the sky is not the same everywhere on Earth. Beneath the colours we see, other traces of the Ice Age also linger in our bodies, written not on the surface but in the ways we process food and manage scarcity. as the story moves from skin to stomach, from sunlight to the chemistry of ancient meals, another layer of this genetic memory begins to unfold. If skin was where the Ice Age sky met the body, then diet was where the landscape itself entered the bloodstream. Every bite of meat, every mouthful of roots, berries, or shellfish was a small negotiation with the environment. Over very long stretches of time, these negotiations left traces in our DNA, hints of the meals that sustained, strained, or starved our ancestors. Across the vast Mammoth steppe which once stretched from Western Europe, through Siberia and into the Americas, life revolved around big animals and the short growing season that fed them. Herds of mammoth, bison, horse, and reindeer grazed on cold-resistant grasses and herbs. The people who followed them relied heavily on meat and fat, with plants as seasonal supplements when they could be found. A large kill meant dense calories packed into muscle, organ, and marrow. Fat, in particular, was precious. It burned slowly in the body, carried fat-soluble vitamins, and helped people through the long, dark months when fresh food was scarce. Far from these inland grasslands, along coasts and river mouths, another Ice Age menu took shape. There, humans learned to read tides and currents, to break open shellfish, to catch fish under the ice, and some places to hunt seals, seabirds, and other marine animals. Their food was still rich in protein and fat, but the profile was different. Marine foods brought large amounts of certain fats, including omega-3 fatty acids and important micronutrients like iodine and vitamin D. While reindeer and mammoth hunters might gnaw frozen marrow in a snowfield, coastal foragers squatted on gravel beaches, prying open shells and stripping meat from bones slick with sea oil. In forests and mixed environments, the menu shifted again. Where trees spread their canopies, especially in slightly milder parts of the Ice Age world or in sheltered valleys, people could gather nuts, seeds, fruits, and starchy roots when the season allowed. They still hunted, perhaps focusing on deer, wild boar, or smaller game rather than huge steppe mammals. But their year had more pronounced peaks of plant abundance. Autumn might bring a rain of acorns or hazelnuts. Late summer, a flush of berries. Underground tubers and rhizomes stored carbohydrates, waiting for those who knew how to dig and detoxify them. Between these worlds, steppe, shore, and forest, lay countless variations. River corridors rich in fish and waterfowl, lake margins with reeds and edible bulbs, rocky uplands where only certain hardy plants and small animals survived. Each local ecology offered a slightly different balance of fat, protein, and carbohydrate, a slightly different pattern of feast and hunger. Small human bands adapted not just in culture and technology, but in the quiet, inherited machinery of their metabolism. Genes do not remember individual meals, but they can bear the marks of long-term pressures, of diets that were consistently heavy in one kind of nutrient and light in another, of environments where famine came often enough that bodies able to store or spare energy had an edge. In such settings, tiny differences in how efficiently someone digested fats or handled sudden surges of sugar could add up over generations. On the Mammoth steppe, where animal fat might be the main winter energy source, people whose bodies could process large amounts of fat without quickly clogging their blood vessels or overwhelming their organs might, on average, survive better. Selection could, very slowly, favour variants that fine-tuned how fats were broken down, transported, and stored. The genes encoding enzymes involved in lipid metabolism, or proteins that ferry fats through the bloodstream, became quiet sites of negotiation. Not for perfect health, but for good enough survival in a harsh, meat-heavy world. In coastal or riverine settings, where diets mixed marine fat, fish protein, and some seasonal plants, slightly different pressures might apply. the specific profile of fats was different. Iodine and vitamin D were more abundant. Certain amino acids and trace elements came in higher doses. The bodies of these people still had to manage scarcity and cold, but the challenges were arranged differently. Over time, the subtle pattern of which variants of metabolic genes were common in these populations could diverge from those on the steppe, each set tuned, if imperfectly, to its own menu. Where forests and mixed environments offered more regular access to starchy roots, nuts and seeds, the ability to extract energy from carbohydrates became more important. Early on, this might have centered on how well the body handled sporadic bursts of starch, how quickly blood sugar was cleared after a rare feast of roasted tubers or ground seeds. Later, after the Ice Age, as some groups leaned more heavily on domesticated plants, the pressures changed again. But even before agriculture, there were differences between groups that relied almost entirely on animal foods for months at a time, and those that could count on periodic stores of starchy plant matter. Metabolism in this sense is not a single trait but a web. How much of an enzyme is made in the pancreas or liver, how sensitive cells are to insulin, how easily excess energy gets packed away as fat under the skin or around organs. Small shifts in this web can tilt the probabilities. Who copes better with a brutal winter on little food? Who recovers faster after a sudden windfall of calories? Whose children survive a lean season? Over time, such probabilities shape the genetic landscape. The local food ecology did more than keep people alive. It shaped who had children and how many. A band that hunted reindeer with great skill but lived in a region where reindeer sometimes migrated unpredictably might face years of plenty followed by sharp crashes. In those lean years, bodies that stretched stored fat a little longer, or that could draw more efficiently on protein without damaging tissue, might carry individuals through a dangerous spring. Along a coast where shellfish were almost always available, but large marine mammals were rare prizes, the pressure might instead favour bodies that could run for long hours over slippery rocks, or stay warm on relatively modest fat intake. These differences were never absolute. People traded, travelled, and sometimes changed their subsistence strategies as climates shifted and new tools appeared. Genes did not rigidly lock them into one diet. But over thousands of years, averages moved. Certain metabolic variants built up in some regions and not others, reflecting in an abstract way the abundance of mammoth fat here, the regularity of acorns there, or the long history of fish and sea mammal flesh along this or that shore. It is important to keep in mind that many of the diet-related genetic variants that draw attention today, such as those influencing how adults digest milk sugar, or how efficiently certain starch-digesting enzymes are produced, either arose or spread much later, in the wake of agriculture and the rise of more stable plant-based food supplies. The Ice Age world was still a forager's world, and many of the stark genetic shifts linked to grains, dairy and settled life had not yet taken place, or were only just beginning in small corners. Still, some of the older layers remain. Patterns in genes related to fat metabolism, to the handling of certain vitamins, or to the storage of energy, hint at ancient episodes of adaptation to different kinds of pre-agricultural diets. These patterns are blurred now by millennia of movement and mixing, yet traces persist. They remind us that our bodies are not designed for one timeless, natural diet, but are products of many changing menus, each leaving faint adjustments in the machinery of appetite, digestion and storage. When we speak today about being well adapted or poorly adapted to some modern way of eating, we are, in a sense, brushing against this long history. A person whose distant ancestors weathered Ice Age winters on seal fat, or followed reindeer across the tundra, or dug roots in cold, damp soils, carries in their cells a set of compromises shaped by those worlds. Not instructions to eat as their ancestors did, but echoes of the pressures those ancestors faced. As the Ice Age wore on, climate swung between colder and slightly warmer phases, coastlines shifted and grasslands advanced and retreated. With each change, opportunities to eat, store and endure were rearranged. Just as skin adjusted to the tilt of the sun, the inner chemistry of the body adjusted to what could reliably be put on the fire and into the mouth. Disease, too, participated in this story, stalking bodies weakened by hunger or scarcity, exploiting the cracks left by hard winters and narrow diets. In the shelter of caves and huts, another dialogue between genes and the Ice Age world unfolded, not about what to eat, but about how to survive the invisible threats that came with cold, crowding, and close contact. Cold landscapes can seem clean in the imagination, stripped of the rot and fever of the tropics. It is tempting to picture the Ice Age as a time when disease pressure slackened, when low human numbers and freezing winds scoured away most pathogens. But the record written in our DNA tells a different, quieter story. Even in the shadow of the ice, life was entangled with microbes, and immunity was under constant negotiation. Pathogens are not only the dramatic killers that sweep through crowded cities, they are also the intestinal parasites that sap strength, the skin infections that flare in small cuts, the respiratory bugs that bloom in smoky shelters, the bacteria that lurk in meat not quite cooked through. In a world of raw hides, shared bedding, and close contact with animal carcasses, there was no shortage of opportunities for infection, even when the air was cold and the human footprint on the land was light. Geneticists can see the imprint of these long struggles most clearly in immunity genes, especially in families like the HLA complex, which helps the immune system recognize foreign invaders. These regions of the genome look different from many others. They are unusually diverse, packed with variants that seem to have been preserved by selection, as if evolution were reluctant to commit to a single solution. The picture that emerges is one of constant shifting battles, where different microbes pushed and pulled our ancestors in different directions, maintaining a kind of biological stalemate. During the Ice Age, these pressures did not disappear, they changed shape. Small bands huddled together for warmth, sharing enclosed air thick with smoke and moisture. In these settings, respiratory infections would have found an easy path from person to person, especially in the deep of winter when outdoor activity shrank and fresh food was scarcer. Wounds from hunting large animals, frostbite injuries, and the incessant friction of clothing and tools against skin could open doors for bacteria that today might be brushed off with an antiseptic wipe, but then could mean prolonged illness or death. Animal contact added another layer. Hunters delved their hands into carcasses, butchered organs, scraped hides, and sometimes stored meat for long periods. Pathogens that moved between animals and humans, zoonoses, would have been part of their world, even if at a lower intensity than in later farming societies. Each exposure was a roll of the dice. Over many generations, lineages whose immune systems were slightly better attuned to the local menagerie of microbes left more descendants. Slowly, the genetic frequencies shifted. One of the most striking pieces of evidence for this continuing struggle comes from the DNA of our archaic cousins. When researchers sequenced Neanderthal and Denisovan genomes, they found that small fragments of their DNA still survive in many living people, inherited from ancient interbreeding. In a number of cases, those surviving fragments are not random. They're enriched in genes related to immunity and inflammation. This suggests that when modern humans moved into Eurasia and met long-established populations, they sometimes picked up useful genetic tools for dealing with local diseases. Imagine newcomers from Africa entering temperate and subarctic zones, already occupied for hundreds of thousands of years by other humans. The viruses, bacteria, and parasites embedded in that landscape had been co-evolving with Neanderthals and Denisovans all along. To the new arrivals, some of these pathogens would have been novel. One way to adapt besides suffering through centuries of trial and error, was to borrow immunity variants from those who were already there, bits of archaic DNA that helped recognize or control these Eurasian microbes were preferentially retained, while others faded away. At the same time, human movement into new environments continuously rearranged the disease map. Rivers and lakes brought waterborne parasites. Coastal zones added marine-associated bacteria and new kinds of foodborne risk. Forest margins harboured ticks and other small vectors. Even shifts in camp location into damp caves, onto windswept ridges, or beside stagnant wetlands would have changed the local pathogen community. Each band, moving through its seasonal round, lived within a distinctive microbial halo, a pattern of exposure that shaped which immune variants were most helpful. These patterns were not only regional but temporal. During colder, drier phases, some parasites that relied on insects may have retreated, while others thrived in stressed animals or crowded wintering grounds. In slightly warmer intervals, when forests regrouped and wetlands expanded, different suites of disease agents could flourish. Our ancestors did not experience one timeless Ice Age disease environment, but a series of shifting combinations. The immune system, with its layered defences, adapted both in the short term, through learned responses in an individual's lifetime, and in the long term, through genetic changes that altered probabilities across generations. Yet every adaptation carried a cost. Many of the genes that help defend against infection are tied to inflammation, the heat, swelling, and cellular alarm bells that help limit microbial spread. A strong inflammatory response can be life-saving in the face of sepsis or a deep wound infected with ancient soil bacteria. But if that same response is easily triggered or hard to shut off, it can damage the body's own tissues. The balance between vigilance and restraint is delicate. Evidence suggests that some variants favoured in high pathogen environments, whether during the Ice Age or later, are today associated with increased risks of autoimmune and inflammatory conditions, arthritis, inflammatory bowel disease, type 1 diabetes, and others. In a world of constant microbial challenge and frequent physical trauma, the benefit of a quicker, stronger response may have outweighed these long-term costs, especially when few people lived into old age. In modern settings, where antibiotics, sanitation, and vaccines reduce many infections, those same variants can become liabilities. Other trade-offs likely involved energy allocation. Mounting an immune response is metabolically expensive. In times of chronic scarcity, bodies that pared back certain immune functions to conserve calories might have had a short-term advantage, especially if their environment carried a relatively low burden of some pathogens. Conversely, in swampy refuges or dense forests where parasites were more common, the scales might have tipped the other way, favouring more robust immune investment even at the cost of growth or fertility. These kinds of subtle shifts would not leave clear historical narratives, but they would leave statistical fingerprints in the genome. There's also a quieter consequence. Adaptations to one disease environment can be mismatched when people move. A band whose ancestors had honed their defences in windswept steppes might relocate over centuries to milder, wetter areas and encounter unfamiliar parasites for which their inherited arsenal was less than ideal. Cultural change, new rituals of cleanliness, different ways of preparing food, the adoption of plant medicines could buffer some of the impact, but the genetic underpinnings would adjust only slowly. Migrations during and after the Ice Age thus layered populations with partly incompatible immunological histories, some of which are still being disentangled in modern medical genetics. In this way, the story of Ice Age disease is not one of absence but of subtle presence. Fewer people and broader distances may have limited some fast-moving epidemics, but they did not halt the constant background dialogue between humans and microbes. The genes we carry today, especially those involved in recognizing and responding to pathogens, are shaped by those long, uneven conversations. The smoky huts in winter, the cut that would not heal, the child who survived a fever because a small change in their DNA tilted the odds. As climates shifted and the ice advanced and retreated, these pressures never entirely let up. They ebbed and flowed, tugging at the genome in different directions, trading lower risk of one illness for greater vulnerability to another, favouring inflammatory zeal here and cautious tolerance there. In the next part of this story, these same forces will intersect with another powerful shaper of our genetic past, the narrowing and branching of lineages as some groups dwindled, others vanished, and a few carried their particular immune legacies through the bottlenecks of the Ice Age and beyond. When numbers are small and the world is unstable, chance becomes a powerful force. In population genetics, there are a few quiet concepts that capture this: bottlenecks, founder effects, and genetic drift. They sound abstract, but they describe things that would have been very real to Ice Age families: the thinning of neighbours, the disappearance of nearby bands, and the fragile persistence of only a few surviving lines. A population bottleneck is what it sounds like, a narrowing. Imagine many human groups spread across a wide landscape, each with its own blend of faces, stories, and genetic variants. Then something happens that sharply reduces their numbers. It might be a long, brutal, cold snap, a drought, a volcanic winter, or an unlucky run of disease and hunger. Most of the lines that have been weaving forward through time are suddenly cut off. Only a small subset of people remain, and from that point on, most future descendants trace back to this reduced handful. A founder effect is a related idea seen from the opposite angle. Instead of a population shrinking in place, a small group moves away or becomes isolated and ends up founding a new, separate population. The variants those few individuals just happen to carry become much more common in their descendants, not because they are better, but because they are the ones that made it into the new branch. It is like copying a massive library by taking only the books that fit in a small satchel. What ends up on the shelves of the new library is constrained by what that first traveller grabbed on their way out the door. Underlying both of these processes is genetic drift, the random rise and fall of genetic variants simply because of who has children with whom, who survives to adulthood, and who does not. Drift is present in every population, but it is much stronger when numbers are small. In a village of thousands, the loss of one family line hardly changes the overall genetic pattern. In a band of a few dozen, a single birth, death, or pairing can push some variants toward fixation and erase others forever. During the Ice Age, climates swung back and forth over thousands of years. Glaciers advanced and retreated. Cold, dry phases pinched resources and broke up habitats. Each swing of the climatic pendulum reshaped the canvas on which human lives were painted. Grasslands could expand into rich hunting grounds for a time, supporting more groups and more genetic variety. Then, as temperatures dropped further, or rainfall patterns shifted, those same areas might become stark, unyielding spaces where only a few bands could hold on. These shifts did not have to be globally catastrophic to carve deep marks into our ancestry. A long period of poor winters could steadily lower birth rates and raise mortality. A cluster of bad years in a crucial region could leave only a patchwork of survivors scattered in pockets, where food, water, and shelter remained dependable. In such periods, the world of our ancestors would have subtly emptied. Trails once marked by frequent smoke and footprints might have grown quiet. Paths between bands could be cut by newly dangerous terrain, frozen rivers, or spreading ice. Isolation was as important as outright loss. When groups were separated by distance, ice, deserts, or simply by a band's decision to stay away from others in hard times, their gene pools began to drift apart. Over long cold centuries, even modest separations could lead to distinct genetic paths. Most of those paths did not lead to the present. Entire lineages, families, clans, whole little nations of related people flowered for a while, left their imprint in the bodies and minds of those who knew them, and then disappeared without leaving direct descendants in today's populations. We know this, in part, because of what ancient DNA has revealed, and, just as importantly, what it has not. When scientists sequence genomes from Ice Age skeletons, they often find genetic patterns that are not cleanly matched by any single living group. It is as if we are seeing ghost branches on a family tree, branches that once held leaves but have long since withered and broken off. These people were not anomalies. They were ordinary participants in their own present. It is only from the view of today, peering backward, that they appear as lost lines. The survival of a few groups through repeated bottlenecks can distort our picture of the past. If one small population happens, by luck and circumstance, to endure a harsh climatic spell while its neighbours vanish, its particular mix of genetic variants becomes disproportionately represented among future humans. Later, when we look back through the lens of modern DNA, it can seem as if those survivors were always central and widespread, when in reality they may have been just one of many regional threads elevated by the accidents of survival. This amplification of a few lines and the erasure of many others means that the genetic story we reconstruct is lopsided. It overemphasizes lineages that happen to pass through the eye of each bottleneck and understates the diversity that once existed in the wider world. Traits that are common today might owe their frequency less to any special advantage, and more to the simple fact that the people who carried them were standing, by chance, in the right valley, on the right side of a glacier, or near the right patch of winter pasture, when the climate tightened the noose. In this view, ancestry becomes a conversation between adaptation and happenstance. Some genetic variants clearly rose in frequency because they helped their carriers cope with cold, with hunger, or with new diseases. Others drifted upward because their carriers were part of a lucky refuge group, or because a particular band had a few more surviving children during a lean generation. Over tens of thousands of years, these small numerical imbalances compounded. The role of the demographic dice, repeated across many bottlenecks, quietly reshaped the human genome. When people today send off a sample of saliva and receive a portrait of their ancestry, they are peering through all of these filters. The percentages and clusters they see reflect not only migrations and marriages, but also the long attritional thinning of lineages under Ice Age skies. The patterns look clean and bounded on a report, but behind them lie generations of near misses, great-uncles who never had children, bands that died out after a bad winter, entire regional populations folded back into others or extinguished outright. To remember this is to soften the edges of identity. The genetic threads you carry survived a long series of bottlenecks, where many neighbouring threads snapped. Your ancestors were, in large part, the beneficiaries of demographic chance, of being slightly upwind of disaster, slightly closer to a sheltering valley, slightly more fortunate in their timing of births and deaths. Those who did not make it through are mostly anonymous now, but they were once as real and central to their own world as we are to ours. In the next part of this story, attention shifts from the narrowing of numbers to the places that sheltered them, the refuges at the edge of the ice where the surviving bands endured, rebuilt, and waited out the cold. It was in those scattered havens that the lineages that would flow into the present were quietly preserved, and where the next chapters of our shared genetic history began to take shape. When the ice pushed hardest into the continents, the world of our ancestors did not simply vanish, it folded inward. As glaciers thickened and sea levels fell, the great plains of Eurasia and North America became harsher, drier, more unpredictable. Many regions that had once supported roaming bands of hunters and gatherers turned into marginal lands where only the most transient visits were possible. In response, people did what living things have always done under pressure. They retreated into refuges. Scientists call these places refugia, from the word for refuge, and they were the sheltered pockets where climate, terrain, and chance combined to soften the worst blows of the Ice Age. They were not tropical paradises, just local exceptions to a brutal rule. Valleys that trapped slightly warmer air, coastlines where sea and land created milder winters, river corridors that stayed rich in plants and game while the surrounding uplands thinned out. In these pockets, human groups could continue to find water, fuel, and food when nearby regions had become too barren or too dangerous to cross. In Europe, the great ice sheets draped over the north forced people steadily southward. Over long millennia, populations that had once been scattered from the Atlantic to the Urals were funneled into a handful of more habitable zones. The Iberian Peninsula, sheltered by mountains and influenced by the Atlantic, became one such refuge. Southern France and northern Spain, with rivers cutting through limestone hills, offered caves, woodlands, and migration routes for animals. Further east, the Italian peninsula, protected by the Alps and surrounded by moderating seas, formed another pocket where human groups could endure cold periods, with a bit more security than those trapped on open plains. The Balkans and the lands around the Black Sea made up yet another refuge zone. Here, the combination of lower latitudes, complex terrain, and proximity to milder seas meant that woodlands and grasslands could persist even during colder phases. To the southeast Western Asia, especially the Levantine corridor of today's Israel, Lebanon, and Syria, and the highlands of Anatolia, acted as a crossroads refuge, linking Africa, Asia, and Europe. These regions did not escape the Ice Age, but they were buffered from its full force. In the worst cold, they became reservoirs of people and genes. Beyond Europe and Western Asia, similar patterns likely emerged. In Central Asia, deep river valleys and areas in the rain shadow of mountain ranges would have served as stepped-back shelters, where small groups persisted while the surrounding steppes oscillated between rich pasture and dust-swept emptiness. In East Asia, coastal plains near rivers and zones protected by mountains from the coldest, driest winds provided relative stability. In each of these places, conditions were not easy, but they crossed some invisible threshold from impossible to barely possible. That difference was enough. Refugia were not just about warmer temperatures. They were about continuity, places where plants could still complete their life cycles, where herds could still find winter forage, where fresh water did not freeze or evaporate away. For human families, this meant that knowledge passed down from parents to children remained useful. Traditional hunting grounds might shrink, but they did not disappear. Paths between seasonal camps might shorten, but they did not all collapse. People could adapt by tightening their circuits, not by abandoning everything they knew. Isolation, however, was the price of safety. As bands withdrew into these scattered havens, contact between them became rarer. Route networks that had once stretched across thousands of kilometers fragmented into shorter, more local webs. Marriages that had once linked distant groups now typically occurred within a much tighter radius. Over generations, this geographic and social narrowing began to leave distinct marks in the genome. Within each refuge, genetic drift and founder effects went quietly to work, amplifying the quirks of whichever families had made it inside. Perhaps one valley in Iberia, by chance, was home to a cluster of people carrying a particular set of variants. Over centuries, as they married, had children, and recovered from lean years, that local genetic signature grew stronger. In another refuge, far away in the Levant or the Italian peninsula, different variants rose and fell under the same quiet arithmetic of small numbers and limited mates. Because movement in and out of these refugees was constrained, new variants arising within them tended to stay local. Mutations affecting everything from immune responses to subtle aspects of physiology and appearance could accumulate within a sheltered population, largely unshared with others. Meanwhile, variants that had once been widespread across Eurasia could vanish in one refuge while surviving in another, simply because the right carriers happened to be on one side of a mountain range, rather than the other when the climate turned. When researchers today look at the genomes of ancient individuals from different parts of Ice Age and post-Ice Age Europe, they can often detect echoes of these separate sanctuaries. There are genetic clusters that seem to trace back to people who rode out the cold in southwest Europe, others that bear the stamp of Balkan or Italian refugees, and still others that align with groups who persisted further east. These signatures are not sharp borders, but they are recurring patterns, like faint dialects preserved in DNA. As the climate warmed and ice sheets began their long retreat, the walls of these genetic enclaves slowly dissolved. Grasslands and forests pushed northward, rivers re-established broad floodplains, and previously glaciated valleys reopened. The refuges did not empty all at once in a grand exodus. Instead, over many generations, people expanded their seasonal rounds, tested new routes, and followed animals into freshly hospitable lands. This outward movement turned refugia from mere shelters into sources. Populations that had been shaped and pruned in isolation now spilled back into the wider landscape. The genetic variants that had accumulated within each refuge were carried outward with them, spreading across Europe and Asia as bands split, travelled and intermarried. Where different refuge-derived populations met, their distinct signatures blended, creating new combinations that would, in time, become the genetic foundations of later regional groups. The process did not happen only once. The Ice Age was not a single freeze, but a series of advances and retreats, pulses of cold and relative warmth that played out over tens of thousands of years. Each major glacial maximum squeezed people back into refuges. Each interglacial or milder phase allowed them to re-expand. With every cycle, populations contracted into sanctuaries, drifted and differentiated within them, then flowed outward again, mixing, spreading, and sometimes being pushed back once more. Across these repeated contractions and returns, a layered pattern emerged in our ancestry. Some genetic variants proved stubborn, surviving several rounds of squeezing and expansion by being present in multiple refugia or by riding along with especially successful groups. Others flowered briefly within a single sheltered population and then faded when that population was absorbed or replaced in a later climatic swing. The genome became a palimpsest, traces of old refuge histories partly overwritten by newer ones, yet still detectable if you look closely. For you, lying far removed from those cold centuries, these patterns are part of why your DNA does not point back to a single place or moment. Instead, it carries pieces of several ancient refuges, fragments from southwestern valleys, from eastern corridors, from southern coasts and upland basins, layered and remixed by countless journeys since. Each of those fragments once sat quite literally at the edge of the ice, preserved in a pocket of survivable world, while much of the surrounding land lay locked, frozen and empty. From these secluded havens, the human story didn't merely endure. It gathered its next momentum. In the following chapters, as climates softened and new corridors opened, those refuge-born lineages would cross rivers and mountains, enter continents they had never seen, and meet distant cousins whose ancestors had taken entirely different routes. The quiet separation of the refugees set the stage for the great re-encounters that would follow. From those scattered refugees, some groups would spend the rest of the Ice Age cycling through familiar valleys and coasts. Others, very slowly, began to lean northward, inching into lands that had once been utterly forbidden. Their story leads, in time, to the threshold of new continents. As the ice sheets slackened their grip and then tightened again in long, slow pulses, vast reaches of northern Eurasia shifted from lethal white deserts to tough but usable grasslands and back again. When conditions allowed, game animals probed the edge of the retreating ice, finding fresh pastures on the newly exposed plains. Human bands, already tuned to read animal movements, followed those shifting herds, always testing the boundary between survivable cold and the kind of cold that kills. This was the beginning of the peopling of northern Eurasia, not a dramatic rush into empty space, but generation after generation of small steps. A family might push their seasonal rounds a little farther north, hunting reindeer or mammoth on summer pastures where, a century earlier, there had been only ice. Children born into these exploratory camps would grow up thinking of those northern forays as normal, and their own children might nudge the route onward again. Over millennia, the frontier of ordinary life crept into regions that had once been unthinkable. Along this frontier, the landscape itself changed in ways that shaped human possibilities. Lower sea levels, caused by the enormous volumes of water locked up in ice sheets, exposed broad shelves along the northern coasts. Shallow seas withdrew to reveal flat plains, braided river channels, and wetlands where birds, fish, and mammals found new feeding grounds. What we now think of as separate pieces of land, Siberia, Alaska, far eastern Russia, were, at the height of glaciation, joined by a continuous expanse of earth and grass known today as Beringia. Beringia was not a narrow bridge to be crossed in a hurry, but a region in its own right, stretching for hundreds of kilometers from west to east. It likely held mosaics of tundra and steppe, patches of shrub and meadow, rivers slicing through the permafrost. For Ice Age hunter-gatherers skilled in tracking large mammals and harvesting wild plants, this was not a frozen obstacle but a new, if harsh, homeland. Groups reaching its western edge from Siberia may first have treated it as seasonal hunting ground, then as regular territory, and eventually as the centre of their world. The existence of such land bridges was a direct, quiet consequence of the ice. As glaciers grew, global sea levels fell, bearing roots that had not existed for hundreds of thousands of years. When the climate warmed and the ice melted back, the seas rose again, drowning those same corridors beneath waves. In between, there was a long window when animals and people could move across what is now ocean, leaving no visible trace on today's map. In the absence of those traces, much of what we know comes from genetics. When researchers compare the DNA of people living today in the Americas with that of populations across Eurasia, they find a pattern that points back to a small set of ancestral groups rooted in Northeast Asia and Beringia. These ancestral lines branched off from other Eurasian populations tens of thousands of years ago, then experienced a long period of relative isolation before spreading through the Western Hemisphere. That pause is one of the most intriguing parts of the story. Instead of a simple model where people in Siberia walk straight into the Americas as soon as a land bridge appears, genetic data suggests something more layered. A group or groups moving into Beringia, then staying there, largely separated from both their Siberian relatives behind them and the vast, ice-bound lands ahead. For many generations, perhaps thousands of years, they seem to have lived in this northern refuge, accumulating their own genetic quirks, adapting to their particular diet and climate, and shrinking and growing with the fortunes of their environment. From a human perspective, that pause would not have felt like waiting to cross into a new continent. Beringia would have been home, a world of familiar campsites along rivers, known herds on predictable routes, seasonal cycles of light and darkness that parents taught to their children. Relations with groups farther west may have continued at low level, through distant kin marriages or rare trading excursions, but the core of daily life was local. In this isolation, drift and selection went to work, giving Beringian populations a distinct genetic signature that still echoes in the genomes of indigenous peoples of the Americas today. Only when conditions changed enough, when ice retreated, when coastlines opened, when plant and animal communities reshuffled, could some of these Beringian groups move farther east and south. The path into the interior of the Americas was not a single open door. At different times, barriers of ice closed the inland route, forcing any movement to track coastal margins. At other times, the ice sheets split, creating a corridor of habitable land between them, which then narrowed or closed again as climate shifted. The genetic evidence, alongside archaeology, hints at a tapestry of movements, some lineages likely following inner corridors, others shadowing the Pacific coast, some pushing deeper, others lingering behind. Every step into these new lands was filtered by the environment. Not every group that reached high latitudes or Beringia necessarily moved on to the Americas. Cold, wind, scarcity of fuel, and the sheer unpredictability of northern climates would have pruned attempts that failed. Families whose toolkits, social networks, or knowledge were not quite matched to the challenges of a particular route might retreat, merge with neighbours, or disappear from the record altogether. Those who did persist and advance carried with them not only their genes but also cultural adaptations, ways of storing food, clothing styles to manage extreme cold, hunting strategies for unfamiliar animals. In this sense, the migration into new continents was less like a single wave and more like a braided river. Different strands of movement started, faltered, split, and rejoined. Some groups branched off into the far northeast of Eurasia and remained there, becoming ancestors of later Siberian and Arctic peoples. Others, after that long Beringian sojourn, moved into the Americas, where they spread astonishingly fast over many generations, adapting in turn to forests, prairies, deserts, and tropical lowlands. Each branch carried only a portion of the ancestral genetic diversity, and each was shaped further by local conditions, creating the genetic patterns we see today. The complexity of these journeys is written in the small details. In mitochondrial lineages that trace back to a specific Ice Age population, in tiny shared stretches of DNA that link distant Siberian and American groups, in signs of ancient bottlenecks where population sizes dipped dangerously low before rebounding. None of these clues alone gives a simple straight line from one point on the ancient map to another. Together, though, they reveal a story of movement that is hesitant, interrupted, and deeply shaped by climate. As you imagine this vast, slow expansion, it helps to let go of the idea of clear borders. For the people living then, there was no label that said Eurasia on one side of a line and America on the other. There were only lands that were known and lands that were not, places where stories and memories anchored you, and places where you might follow the tracks of an animal until the world became strange. The continents, as we name them now, were for them a continuous, if uneven, surface of stone, ice, soil, and water. What makes the crossing into new continents feel so momentous to us is that, from our vantage point, we can see the whole arc, small bands emerging from Ice Age refuges, edging north along widening plains, settling into Beringia, then, after long waiting, spilling into entirely new ecosystems. But lived from the inside, it was a chain of modest decisions, each one bound to the next season's needs, where to find caribou, where the salmon would run, where the snow crust would bear your weight. The grand sweep of peopling the North and the Americas was built out of those quiet choices, made again and again under cold skies. In your own DNA, if you carry ancestry tied to these roots, the legacy of that long, gradual process survives as a handful of markers, faint but enduring. They do not tell of a single heroic crossing, but of many winters endured, many cautious experiments with new ground, many families that learned, by trial and memory, how to live at the shifting edge of the ice and then beyond it. For most of this story, we have followed the line traced by genes. Yet the way those lines are drawn is itself a quiet, painstaking process, full of limits and uncertainties. Before we let DNA carry us further, it is worth pausing to ask what it can truly reveal and what it cannot. Ancient DNA begins as dust and fragments, in caves, in frozen ground, in the dense cores of teeth and the inner chambers of bones. Tiny threads of genetic material can survive for thousands, sometimes tens of thousands of years. They are not pristine scrolls of information, laid aside for us to read. They are more like scattered letters from a book, mixed with soil, microbes, and the genetic traces of everyone who has handled the remains since they were found. Recovering those letters is an exercise in caution. Researchers shave off thin layers of bone or drill into a tooth, often from the densest part near the root, where DNA is best sheltered from time. In laboratories designed to keep out modern contamination, they coax what is left of the ancient molecules into revealing themselves, then use sequencing machines to read the raw code. What comes out at first is a blur, a jumble of short, damaged stretches of DNA that must be sorted, aligned, and compared to reference genomes from humans, other animals, and the teeming communities of microbes that also colonize bones. Time leaves a fingerprint on DNA. The old strands are broken into short pieces, and certain chemical changes, like the gradual swapping of one base for another at the ends of fragments, are more common in ancient material than in modern. By looking for these patterns, scientists can tell which bits of genetic code are likely to be truly old and which are more recent intruders. In this way, damage itself becomes a tool, a subtle test that helps separate Ice Age whispers from the loud noise of later life. Even with these tools, contamination is a constant concern. Every modern human cell that touches a bone, every breath, every brush of skin, adds fresh DNA that can overwhelm the faint signal of the past. Early attempts at ancient DNA were often clouded by this problem, leading to confusing and sometimes misleading results. Today, strict cleanroom conditions, protective clothing, and careful controls help to reduce that risk, but it can never be removed entirely. Each sequence must be examined for telltale signs of intrusion, unusually long fragments, or patterns that match lab workers more closely than any plausible ancient population. Preservation, too, is uneven. Cold, dry, stable environments tend to protect DNA. Hot, wet, or highly variable conditions destroy it. This means the Ice Age world is not equally visible to us everywhere. Permafrost, high caves, and sheltered burials in temperate zones may yield genomes in surprising detail, while vast stretches of tropical and subtropical land, where humans also lived and moved, offer far fewer preserved genetic traces. Our picture of the past is shaped not only by where people went, but by where their molecules have survived long enough to be read. Sampling adds another layer of distortion. Each ancient genome we recover comes from a particular person at a single point in time, in a specific place. For many regions and periods, we have only a handful of individuals or none at all. It is as though we're trying to reconstruct a long story from scattered, incomplete pages, most of them torn from different copies of the book. We can see patterns emerge when enough pages are gathered, but we must remember how much is still missing, and how easily new finds can shift the outlines of the tale. Because of these gaps and biases, ancient DNA is at its strongest when it is woven together with other kinds of evidence. Archaeology gives us tools, dwellings, food remains, and burial practices, traces of how people lived, what they made, and how they treated their dead. Climate records, locked in ice cores, lake sediments, and fossil pollen, tell us when glaciers advanced and retreated, when grasslands opened or forests closed in, When genetic signals of population change are lined up with these environmental and cultural records, the story becomes more grounded. A shift in ancestry at a site gains context if it appears just as a new tool style spreads, or when a cold snap forces game animals to move. Without that context, genetic data alone can tempt us into oversimple stories. A change in DNA through time at a location might reflect incoming people, replacing earlier groups, but it might also show intermarriage, or small bands merging, or even just a few individuals from elsewhere becoming unusually influential in a community. Similarly, a shared genetic marker between distant populations does not, on its own, tell us what language they spoke, what myths they told, or how they understood themselves. It only shows that at some point their lineages were entwined. This brings us to the quiet distinction between ancestry, culture, and identity. Ancestry, in genetic terms, speaks of shared descent, of lines of parents and children stretching back through time. Culture is learned. The skills, beliefs, symbols, and habits pass from one person to another, not by blood, but by teaching, imitation, and shared life. Identity is how people see themselves and are seen by others. Their names for their groups, their sense of belonging, the stories they tell about who they are. Ancient DNA can illuminate ancestry. It can show that two distant groups share forebears, that a population shrank and then regrew, that new genetic strands entered a region at a particular time, but it cannot on its own tell us what those people called themselves, whom they considered kin, or how they divided the social world into us and them. People with very similar genes can belong to very different cultures. People who are genetically diverse can share a common language and way of life. Over the millennia of the Ice Age, adoptions, alliances, captives taken and integrated, and wanderers switching groups would all have blurred any simple mapping from biology to belonging. The same caution applies when we think about traits and behavior. Some genetic variants are clearly linked to physical adaptations we have mentioned, the way the body handles fats, or responds to low vitamin D, or copes with cold. But most aspects of how people acted, thought, and organized their societies do not leave neat genetic fingerprints. No ancient genome can tell us how brave someone felt on a winter hunt, how gentle they were with children, or how they resolved disputes. The temptation to tie complex behaviors, like aggressiveness, creativity, or social structure, directly to genes is strong, but history warns us against it. There is also the danger of reading our present categories back into the deep past. Modern ideas of race, shaped by recent centuries of politics and inequality, do not map cleanly onto Ice Age populations. Back then, small, shifting bands mixed and separated in patterns utterly different from today's nations and continents. To imagine rigid, colour-coded blocks of humanity stretching across the glacial world is to misunderstand both the genetic evidence and the lived reality of those times. So as we listen to what ancient DNA can say, we hold at the same time an awareness of its silences. It tells us about connections and separations, about bottlenecks and expansions, about long ago meetings between different human kinds. It helps us see how the cold and the land shaped the flow of people. But it does not speak alone. It must sit beside stone tools, hearths and middens, cave paintings and carved figurines, pollen grains and ice bubbles. When these lines of evidence are read together, patiently and without haste, they offer a richer, more humble picture. The Ice Age becomes not a simple genetic puzzle to be solved once and for all, but a layered living history in which biology, culture, and climate are constantly entwined. In the quiet of that realization, we can turn, more gently, to the question that touches the present most directly: how the echoes of those old adaptations and journeys still flicker in our bodies and lives today. If we listen closely, the Ice Age is still speaking in the present, not through mammoths and glaciers, but through living bodies. Every day, without our noticing, old adaptations move quietly through modern lives, sometimes helping, sometimes hindering, always reminding us that we are the descendants of people who survived a harsher world. Some of these echoes are clearest when we look at how different populations respond to the same modern conditions. Take, for example, how our bodies handle sugar and fat. For many Ice Age groups, reliable surplus food did not exist. Meals were irregular, seasons were starkly different, and a missed hunt or a failed gathering could leave people hungry for days. In that world, genes that encouraged the body to store energy quickly and efficiently were useful. A metabolism that hoarded calories, that held on tightly to fat in times of plenty, could be the difference between life and death, when the cold set in or game migrated away. In much of the modern world, we have reversed the problem. For many people, food is abundant, energy-rich, and constantly available, even late at night under artificial lights. The same tendencies that once helped our ancestors survive winter scarcity can now push us toward obesity, diabetes, and heart disease. What we call lifestyle diseases are in part tensions between ancient biology and a new environment that changed too quickly for genes to keep up. The Ice Age is not gone. It lingers in how our cells still expect lean months that never come. Vitamin D is another thread tying present to past. In high latitudes, under low winter suns, Ice Age people faced long periods with little ultraviolet light. We have already traced how skin lightened in some lineages to let more sun pass through, allowing the body to make enough vitamin D for bones and immune function. Those adaptations have not disappeared. Today, populations whose recent ancestors lived near the equator often carry darker pigmentation well suited to intense sunlight. When families with that inheritance move to cloudy northern cities or spend most days indoors, the combination can lead to vitamin D deficiency unless diet or supplements make up the difference. On the other hand, people with very light skin living in regions with strong sun face a different Ice Age legacy, a higher risk of skin damage and cancers because their ancestors were selected in dimmer climates. The same trait that helped gather scarce light in glacial Europe can become a vulnerability on a bright beach or a high plateau. Neither set of genes is better or worse in any absolute sense. Each is simply tuned to a different patch of the ancient map, now carried far from its original setting by ships, trains, and planes. Immune systems, too, bear the marks of old environments. During the Ice Age, as we saw, humans in some regions met new pathogens while clustered in cold weather shelters or crowded refugees. In other zones, like parts of the tropics, people faced a very different disease landscape. Heavy loads of parasites, fevers, and insect-borne infections. Over thousands of years, genetic variants that helped resist local diseases were favoured. Some of those variants persist in modern populations even when the disease that once selected them is gone or greatly reduced. This can have complicated effects. A variant that once helped fight off a particular infection might now predispose people to autoimmune conditions, where the immune system turns its intensity on the body itself, or a gene that helped clot blood quickly in case of injury during a winter hunt could, in an older person sitting long hours on a plane, increase the risk of dangerous clots. These are not moral judgments written into DNA, but practical mismatches between old adaptations and new ways of living. Through all of this, one thing becomes clear. Modern health cannot be cleanly separated from deep ancestry. Yet the story is not as simple as mapping traits onto single, pure races or tidy ancestral boxes. Over tens of thousands of years, migration and mixture have woven human lineages together in ways that defy sharp boundaries. The ancient world was full of movement, small bands splitting and rejoining, following herds, exploring new valleys, returning to old refugees, and sometimes meeting and mixing with distant cousins. Those patterns did not stop when the ice retreated. They accelerated. Agriculture, seafaring, empires, trade routes, and forced migrations in more recent centuries layered population on population, until nearly every person alive today carries strands of DNA that originated in many different regions and climates. When geneticists look at modern genomes, they do see patterns that roughly correspond to geography. Certain variants are more common in people whose recent ancestors lived in specific areas, but the edges are always blurred. There is no sharp line on the map where one kind of human ends and another begins. Instead, there are gradients, overlaps, and countless tiny threads of shared descent connecting coast to inland, north to south, and continent to continent. This is one of the greatest gaps between popular ancestry myths and what population genetics actually shows. Stories of bloodlines often imagine straight, unbroken paths. Pure, this, full, that, a single origin carried down unchanged. The genetic reality of Ice Age and post-Ice Age humanity is almost the opposite. Our lineages branch, rejoin, bottleneck, and spread out again. They pick up pieces from neighbours, from travellers, from captives, and from wanderers welcomed in. There is no single tribe that owns survival. Everyone alive today descends from many groups, many refugees, many moments of both loss and resilience. Even traits with clear geographic patterns, like lactose tolerance, or some skin and hair types, or certain cold weather adaptations, are not the markers of neatly bounded ancient races. They are local responses to local problems, the need to digest milk in herding societies, to balance sunlight and vitamin D, to stay warm and still move freely. Those solutions spread along paths of marriage, trade, and migration. Over time, they mixed with one another, so that a person might inherit a cold-adapted variant from one ancestral stream and a malaria-resistant one from another, both quietly working in the same body. This means that everyone, whatever labels their society uses, carries a history of movement under the skin. Perhaps some of your genetic variants once helped a small band survive in a frozen valley. Others may have travelled through humid forests, or along coasts rich in shellfish, or across wide grasslands. Each of us is a convergence point, where multiple ancient journeys meet. The Ice Age is not a distant, separate chapter. It is one of the foundations on which our present diversity rests. It also means that modern identities, national, ethnic, cultural, are poor guides to deep genetic structure. Two people who both call themselves by the same name today might carry quite different mixtures of ancient lineages. Two others, who see themselves as belonging to very different groups, may share surprising threads of ancestry if we trace their genes back far enough. The stories people tell about belonging are shaped by language, memory, borders, and recent history. The stories inscribed in DNA are shaped by climate, chance meetings, and the quiet arithmetic of births and deaths over tens of millennia. Holding these two kinds of stories together requires gentleness. Ancient DNA can remind us that our bodies are archives of old climates, old diets, old diseases. It can also remind us that no one is a simple descendant of a single Ice Age people. We're all products of mixture, of narrow escapes through bottlenecks, of lineages that vanished and others that endured. As you lie here, breathing in a world without mammoths or miles of ice at your door, your body still carries solutions to problems you no longer face, and traces of journeys you never walked. The chill winds of long-vanished winters shape the genes that help regulate your sleep, your metabolism, your immunity, even your bones. The grasslands and refuges of a broken, glacial world still whisper in every cell. Soon we will turn more directly to that idea, that under the skin, without a word from you, the long memory of the Ice Age is always there, quiet and patient, folded into who you are. The Ice Age does not live in us as a single clear story. It is not a movie playing in high resolution somewhere in your cells. What your DNA carries instead are fragments, a change in a protein here, a stretch of Neanderthal sequence there, a tiny shift in how your body handles fat or fights infection. Each fragment is like a page torn from a much larger book, surviving by chance while most of the text has been lost. When scientists read those pages, they can sometimes glimpse the shape of the missing chapters. A cluster of cold-adapted genes hints at ancient winters. A signal of mixture points to a meeting between distant bands. A long, unbroken segment of DNA suggests a recent ancestor. A short, scrambled one tells of deeper time. Yet even with all our tools, most of the human past remains a constellation of glimpses rather than a continuous line. Across this scattered record, a few themes stand out clearly. One is climate. For tens of thousands of years, ice advanced and retreated like a slow tide, reshaping coastlines, carving valleys, and redrawing the borders of forests and grasslands. Human bodies bent in small genetic ways to these changes, skin tuned to different levels of light, metabolisms adjusting to different kinds of scarcity, immune systems practicing against different sets of threats. No single change tells the whole story, but together they form a quiet map of how people learned, generation by generation, to survive on a restless planet. Another theme is movement. People followed herds, tracked rivers, skirted ice sheets, and crossed narrow seas. They climbed into new highlands and slipped through mountain passes. Sometimes they left traces in stone tools and hearths. Sometimes they left only their descendants, carrying a few genetic markers of a journey long forgotten. Migration was not a single wave, but a series of overlapping ripples, out of Africa, across Eurasia, into the Americas, back again, sideways, around and through. Your DNA does not preserve the roots in detail, but it remembers that moving, splitting, and rejoining were the human norm. A third theme is encounter. As modern humans spread, they met other humans, Neanderthals in western Eurasia, Denisovans farther east, and perhaps other groups we've barely begun to name. Most of these lineages vanished as distinct populations, but not entirely. Some of their genes were carried forward, sifted by natural selection and chance, and now live on in people today. You may carry a handful of their sequences without ever knowing precisely where they came from, like old notes copied into a newer book, their original authors lost, and then there is chance itself. Storms, plagues, a single winter that was slightly too long, a crossing that almost failed. So many turning points depended on accidents. A small group might, by luck, carry a rare helpful variant into the future. Another might disappear, taking unique lineages with it. Bottlenecks, founder events, random drifts in gene frequencies. These quiet forces shaped ancestry as surely as climate and adaptation did. DNA preserves the outcome but rarely the reasons. It shows us which lineages endured, not why others did not. When you bring these themes together, climate, movement, meeting, and chance, the result is the deep ancestry of everyone alive now. There is no one line that leads straight from a single Ice Age campfire to you. Instead, there are many lines, weaving and crossing, sometimes thinning to a few fragile threads, and then thickening again. You're not the end of their story, only the current point at which they all happen to meet. This can be a comforting thought. Lying here in the present, surrounded by technologies and landscapes your Ice Age ancestors could not have imagined, you are still connected to them in every heartbeat and every breath. The way your body handles the dark of night, the chill of a cold room, the warmth of a meal, or the scratch of a healing wound. All of these responses are shaped by histories that extend back through glacial valleys and open steppes, through shelters of bone and hide, through snowfields lit by faint distant stars. You do not need to know the exact paths. You do not need to trace every ancestor or identify every mutation. The continuity is there all the same. Their struggles against hunger and cold, their careful sharing of food, their patience through long winters, their choices to move or stay. These are folded in subtle biochemical ways into how your body works while you simply rest and sleep. At the same time, our understanding of this long memory is incomplete and always changing. Each new fossil, each newly sequenced genome, each improved method of analysis adds a few more lines to the scattered pages. Sometimes discoveries confirm what we suspected. Sometimes they overturn comfortable stories and force us to redraw the map once again. Whole populations once thought to be dead ends turn out to be ancestors after all. Genes assumed to do one thing are found to do another. Humility is the only steady position in the face of such a vast, partly hidden past. The Ice Age world that shaped humanity was too large, too varied, and too full of unrecorded days for us to ever reconstruct it perfectly. DNA, for all its power as an archive, is a partial witness. Archaeology, climate science, and anthropology, too, can only approach the truth in pieces. Much of what really happened, the sound of particular voices, the feel of a certain winter morning, the thoughts of a parent watching a sleeping child in a cave, will always remain beyond our reach. And yet, even in this incompleteness, there is a kind of peace. You do not need the full story for it to be real. The long memory under your skin is not a list of names and dates, but a quiet continuity of survival. It is enough to know that you belong to a chain of life that endured ice ages and interglacials, droughts and floods, eruptions and long calms, narrow escapes and lucky abundances. As you drift towards sleep, you can imagine that chain not as a strict line, but as a wide, branching river of ancestry, flowing through deep time. You're one small eddy on its surface, one moment in its long course. The ice has come and gone. The mammoths have vanished. The grasslands have shifted. But something in that river kept moving forward, carrying with it fragments of memory in the language of genes. Tonight, you share your rest with all those unknown ancestors, not as ghosts or distant heroes, but as quiet presences in the way your cells repair themselves, the way your lungs take in air, the way your heart settles into a slower rhythm. Their world is gone, but its lessons remain, written in a script your body reads without effort. The research will continue. New findings will refine and rearrange the picture. Future generations will see connections we cannot yet imagine. Still, the essence will be the same. Humans adapting, moving, meeting, and enduring on a changing Earth. For now, you can let the question soften and fade. The long memory under your skin is still there, patient and unhurried. It does not need to be fully understood to hold you. It is enough in this quiet moment simply to lie here, breathing as the ancient world rests within you, and the present world settles gently around you like falling snow.